Okay, let's start. Or do we have any questions about anything before I show you your cheat sheet for the exam? Any questions about anything? There was a problem yesterday afternoon with the grades or with being able to see them. As of 9 o'clock last night, I could see them again. And so hopefully that won't happen again. I mean, I lost, from my view, there were no grades and there were no quizzes. Okay? Questions about anything? Okay, so in the past, the way I've always sort of summarized metabolism is to basically go over the reactions that you need to know and let you produce basically a sheet you can bring with you. That way I'm doing an online section, so that's hard to do, and then I thought, well, no, I can still do it and just put it up. So we're going to start today, we're going to finish today with, and I'm sure I'm going to forget a few things, but basically what you should know are the pathways, where they're located, what goes in and what comes out, and the important intermediates, the byproducts. So water is not a product in most reactions. Actually, the only place where water is an important product is in the terminal oxidase step, where oxygen is reduced to water in the mitochondria. And then the other step where water is actually involved that's important, of course, is when water is oxidized in PS2 to produce the electrons needed to run photosystems, and then the useless byproduct of that is oxygen, okay? So let's start up here at the CO2. I'll put two. You should be seeing something now. A really bad drawing, okay? And I'm going to do this. Oh! I keep remembering when I hit the thing on the screen doesn't work. Thank you. And you'll have to blip on several times. You know, it used to be set up when you logged on, all this stuff just happened automatically. Now it's slowly failing over time. Is the and the screen's all the way down? Okay, so let's take a CO2 through the stomates. And what we're going to do is we're going to do C3 photosynthesis. So the first thing that happens is the CO2 has got to go into the stomate and it has to get into the chloroplast, okay? And the first reaction, CO2 is combined with a 5-carbon, right, to produce a triose phosphate. Okay? And that's the reduction phase of photosystem, okay? Hopefully you can make your drawing better than I can. Then, I'm sorry, that's carboxylation. I'm getting ahead of myself. Then we have reduction. Oh, good. So we have CO2 into the chloroplast, and we have carboxylation to the triose phosphate. Then we have reduction, right? Where we have the light, ATP and NADPH. From the light reactions that are used to end up with another form of triose phosphate. And then the rest of these really complex reactions are just to regenerate the 5-carbon compound that then combines with CO2 to start the cycle again. Okay? So now let's make this the chloroplast. What moves out of the chloroplast? For the most part, we got two things. The main thing that moves out, remember? No starch, triose phosphate. And every time a triose phosphate moves out, a phosphate moves in, right? Balance the phosphate, or we'd run out of phosphate. Okay? So most carbon moves from the chloroplast into the cytosol as 3-carbon compound, 3-carbon phosphorylated compounds. However, we now know that we can also get glucose coming out of the chloroplast too. Okay? That's a newer finding. So, what happens to the triose phosphate? Let's burn the carbon. We've just made the carbon. Now let's take it back to CO2. What happens next? Anybody remember? Let's go up here and let's say we're going to go with glucose. Glucose is going to be phosphorylated.
Where did the glucose come from?
The chloroplast.
From what?
Oh, inside the chloroplast, glucose can be synthesized from three carbon compounds inside the chloroplast. Okay, that's the actual production of glucose in the chloroplast is a fairly new finding. But what I'm going to do is now I'm going to start glycolysis with glucose. Okay, so I'm taking the and this glucose, of course, can come from anywhere. So we add an ATP, we add another ATP, and then which ends us up with fructose 1,6-diphosphate, and that fructose is split into 2 3-carbon compounds, which is, this is important, where most of the triose phosphate enters, okay? So if you think about it, metabolically, in the leaf cell, it doesn't make any sense to make glucose and then break glucose down to the triose phosphate. So this triose phosphate is coming out of the plastid, and then we go through a series of steps where we have NADH made, and then very importantly we have ATP made. What you should know is that this ATP is made by substrate level phosphorylation. It's not by electron transport. It's made. That reaction requires ADP as a substrate. So we end up with ATPs. I did not ask you to balance these things, but just to show you, we have an ATP here. We have an ATP here. Then that gives us this fructose 6-phosphate compound, which is split into 2. So now we have 2 NADPHs produced for every glucose. We have 2 ATPs here. We have 2 ATPs here. And we get down here to the product, which is pyruvate. Okay? This is why plants can live with glycolysis alone. Count the ATPs. 2, 4, 1, 2. We use 2. We make 4. So we net 2 ATPs. So the plant can survive, not thrive, with just glycolysis. But what else must be active in order for this pathway to work? Nope, not the Krebs cycle. what other thing needs to work to keep glycolysis turning? Oh, come on, folks. Right? And how do you do the NADH to NAD? How do you produce NAD? Oh, come on. Fermentation, right? Pyruvate loses the CO2 to produce an acetaldehyde, which then produces ethanol. And when this occurs, this NADH is cycled back to NAD. Okay? Let me change to another color here. So this cycles NAD back. So the purpose of fermentation is, like I think I put in one of your questions, not to keep Northgate alive. The purpose of fermentation is to cycle NADH to NAD. Okay? Yes, okay. Let's back up here and let's talk about our other photosystem. The other C4. And let me just... Okay, remember, the take-home message is all plants have the C3 cycle. And now let's turn this plant into a C4, which has two kinds of cells and two very distinct kind of chloroplast. We have the mesophyll cell, where the CO2 is fixed by PEP carboxylase into a C4 organic acid. Okay? PEP carboxylase. It's carboxylator.
So, are you saying the C3 photosynthesis ends at fermentation?
No, we're back over here. We finished fermentation. Now I'm talking about C4. Okay? So I started off with, I need some more colors. I started off with a normal C3 plant and take it through to pyruvate, and we're going to talk about what happens under oxygen in a bit. Without oxygen, pyruvate is burned to ethanol. Now I'm backing up, and we're going to talk about C4s. Okay? So the take-home message that you should know is that all plants have the normal C3 metabolism, if you want to call it the normal metabolism. With C4s, where C4 plants found? Where there's a tremendous amount of water and light, so they got a lot of energy. And so what they do is they fix the CO2 initially into a C4 organic acid, which is then moved into the bundle sheath cells, where it's decarboxylated, and now it enters the normal C3 the normal C3 photosynthesis. That's not correct to say normal, but then it goes through C3 photosynthesis, okay? And the purpose of this is to reduce, or actually eliminate, oxygen from being fixed, right? You know, there's a diagram that I've shown you probably 5 or 6 times through the semester of a C3 plant, and as you increase the concentration of oxygen, the concentration of the activity of photosynthesis goes down, because oxygen competitively is a competitive inhibitor. So what happens with the C4 is that oxygen does not interfere with PEP carboxylase. So there's the oxygen there's no worry about the oxygen here. And then in the bundle sheath cells, remember these are the cells in the center, the CO2 concentration is so high that oxygen never enters into the reaction. So C4s never fix oxygen. C3s fix oxygen maybe 1 every 4 times. So think about that. In your normal C3 plant, you're losing a lot of efficiency because of oxygen. Okay? What am I missing before we go on to the next page? I'm not going to try to draw this on one page. So we've covered C3, C4. We've covered moving the triose out of the plastids and having that same cell then put that triose through glycolysis to produce pyruvate as the end product. Okay? So let's take pyruvate. Why does it not let me go to the next slide? I'm going to have to erase this whole thing. What is going on? The clear is not working. I'm going to have to erase it, I guess. Okay, let me just exit out and start it again. Okay. Now let's put pyruvate up here at the top. Now let's talk about what happens in us and in plants that have oxygen. Okay? First thing that happens is pyruvate loses a CO2 when it enters in the mitochondria. Okay? Where then we have a series of reactions where we have the acetyl-CoA, and you don't need to worry about the acetyl-CoA, is converted into CO2. I'm sorry? The pyruvate loses a CO2 to become acetyl-CoA. Okay. That's where the first CO2, you don't have to worry about acetyl-CoA, I'm just telling you that. Then it goes through a series of reactions where this 3-carbon pyruvate ends up being completely burned. Here's a CO2 there, here's a CO2 here, here's a CO2 here. And in these reactions we have NADH made. NADH. We also have an ATP made by substrate level phosphorylation. And we also have and you don't need to worry about the FADH2. Okay? So, that then is what goes on in the center of the mitochondria. And we've produced a little tiny bit of ATP, and we've produced a lot of NADH. Okay? So, what happens next is the NADH it's supposed to be a membrane. The NADH then enters electron transport. Okay? And it goes through the different complexes, and as it does, by the movement, it's going to generate protons, and just like, I've got them on the wrong side, and just like we saw in the chloroplast, those protons move through the ATPase, and as they do, they produce ATP, okay? A lot of ATP. In fact, if you do the counting, if we burn a glucose molecule, we produce about 36, 38 ATPs. That's a lot of energy that we produce. Yes, ma'am.
For the electron transport chain,
Uh-huh.
on the actual slide, it says NADH plus H moves through this chain.
Right.
So which one is like the one that makes the proton gradient, and then which one, where does the electron come from?
Okay. Well, when you see NADH plus H, you're actually the electrons go with the protons. They just don't put them there. So the electrons are actually what's actually moving through the systems, and then the protons are moved. Then are what generate the gradient. And I started drawing it upside down.
And
Okay.
they both come from NADH?
Right. So the NADH is oxidized. It loses its electrons, and you're left. And then those electrons fire all this off. Now, what is the one thing that plants need to keep this system going? Oxygen. We have the terminal oxidase. So 99.99% of why plants and animals use oxygen is to keep this going. Those electrons, O2, those electrons reduce oxygen to water. Okay? That's why we need oxygen. We do have oxidases and other things, but. And so this terminal oxidase is also inhibited by what? Not inhibited by oxygen. Oxygen is a normal substrate. What looks like oxygen? Cyanide. Okay? Okay. What am I leaving out? Oh, yeah, I am leaving out something. Plants have cyanide-resistant respiration, which was a really bizarre finding because cyanide knocks the mitochondria out. Okay? But what we've found over time is that back in close to the start of electron flow, there is let me draw it up here, an alternate oxidase. Which takes oxygen and converts it into water. Okay? And it's not affected by cyanide. But because of its location, there's not much ATP made. So it's located anywhere between the first and the second, or maybe the third one. So you don't get this tremendous generation you don't get all the electron flow, so you don't get as many protons produced, so you don't get as much ATP produced. Okay? And the reason for the alternate oxidase, it's not completely, we know some functions of the alternate oxidase, but we really don't know the reason. Some functions of the alternate oxidase, and I always like to give an animal example of this. Back at the turn of in the 1900s, late 1800s, 1900s, you could buy a really efficient diet aid that was low concentrations of cyanide. So if you have low concentrations of cyanide, you're going to block this step here a little bit. So that means you don't produce as much ATP. So we need more ATP So our cells respond by speeding up metabolism, moving more glucose through glycolysis, producing ATP, which results in heating up a little bit. But what you're doing is you're pumping a tremendous amount of glucose through glycolysis, i.e. you're going to lose weight because you're burning a lot of sugar. Okay? And if you take a little bit too much, you die. So the same thing happens in plants. And the classic one, and the one that's in your book, is something called skunk cabbage, which is this huge plant that is pollinated usually when there's snow on the ground even. And so what happens to a plant when this alternate oxidase happens, so with the alternate oxidase, you're still using oxygen, but you're not producing as much ATP. The plant responds by speeding up glycolysis and heating up. It gets hot, gets warmer. And as it gets warmer, you can imagine why it's called skunk cabbage. As it warms up, the volatiles are produced and the plant's pollinated. Okay, and there are a few other real-world examples of when the alternate oxidase seems to be used. So in this case, this plant, skunk cabbage, and there's more than a few people working on it because they're trying to figure out how it decides to kick in the alternate oxidase. What does it do? And in some cases, some plants, it seems like that, remember we talked about, and you're not going to have secondary metabolism, but we talked about cyanoglucosides, that they may actually be able to split the glucose and the cyanide, and they may actually poison their own mitochondria to increase the alternate oxidase. The alternate oxidase is not something you kick in and then kick off and go back to normal. So the alternate oxidase is sort of like ethylene. You turn it on, the plant's pollinated, the seed's made, the plant's history. Okay? Am I leaving anything out? Yes, ma'am.
So the electron transport chain and the Krebs cycle, where are those? I know they're in the mitochondria, of course.
The Krebs cycle is in the matrix, or the TCA cycle, and electron transport's in the membranes. And if you squint your eyes and look at electron transport in mitochondria, it looks a heck of a lot like a chloroplast, which means in all probability the chloroplast and the mitochondria were at one point the same endosymbiont. And then over time, first came apparently the development of primitive chloroplast, which resulted then in blue-green algae. I think were the earliest ones. Anybody know? Is it blue-green or the first ones? Cyanobacteria, which slowly started increasing the concentration of oxygen. Oxygen was a useless byproduct. And then over eons, as the oxygen concentration raised, then the other systems started separating out.
Yeah, they made that discovery in lichen, because it's that's the mutualistic relationship between the cyanobacteria and fungus.
Okay.
They made that discovery, and I can't remember when it was, but it was because they found something in lichen that they were like, oh, okay, well, all right.
Yeah, they did some experiments in the 50s where they were taking inorganics and zapping them with lightning in a lab with an electrical current, and you could actually end up getting amino acids. And then if you kept doing it, you'd get more complex amino acids. I think it's about as far as I got. They didn't make anything. Okay, am I leaving anything out? So this is your cheat sheet. Okay? Hopefully you can draw better than I can. And that is the level, mainly, this is the level of detail that you should know. Okay? Not how many, not the location of NADHs produced in mitochondria, but that they're produced. That the mitochondria also produces ATP by substrate level phosphorylation. Do you understand that? That's not by proton transport. Substrate level means for that reaction to occur, ATP is made. So in glycolysis, it's incredibly important because without oxygen, that's all the ATP the plant has. CAM. I didn't miss CAM. Okay. And I just erased it. Okay. CAM, not as efficient, and basically it's C4 at night and C3 in the day. The big difference with CAM is that the organic acid is stored in the vacuole. And we had a project years ago where we were making wine from prickly pears, the really the reddish ones. So we found if we picked them at night, they actually had a much lower pH because the organic acids were being made. And so it makes a beautiful wine. Wine is a Star Trek fluorescent color. It's a beautiful, has a great nose. And if you picked them during the day, the pH was a lot higher because the organic acids had already been put through the C3 cycle, okay? So you can measure the vacuole of a cactus at night, and the pH is going to be much lower because it stores organic acids. Anything else I'm leaving out? CAM, C3. Sure? Before I wrap it up, any other questions? So we have one more class on Monday?
You don't have to redefine class on Tuesday?
No redefined class on Tuesday. That's just too weird. Okay, good luck. You know, I had my first hangout last night in HEB. I had 2 people on my phone asking me questions. That was pretty cool. Actually, I've had more people talk to me on their phones than I have actually gone online. People use their computers. And also there's... Do what?
We do have class on Monday.
They have class on Monday. Yes, ma'am.
Is the test due on Sunday or Monday?
I want to say it's due after Monday. Go look. You can look at it and it'll tell you when it's going to be due.